tree phenomenon is depicted on this figure here, and blatantly using some very cheap parlor tricks by not showing you what the axes are, not labeling it. But really what I'm trying to do is pique your interest and try to sort of simulate some of the fascination that I've felt about this biological phenomenon since long before I started studying it formally. This is a hibernation trace of a Colombian ground squirrel, specifically the body temperature. I collected this data with a little temperature-sensitive collar you can see tucked up under this animal's chin. She received her collar on the 20th of August, started hibernating a couple of days later, and then spent the majority of the next 8 to 9 months essentially paralleling the ground temperature. Periodically arising to a warm, euthermic 37-degree body temperature, about what we're all sitting at right now, but for the most part existing with a body temperature a little bit above the freezing point of water. So this time, exactly 1 year ago today, the squirrel had a body temperature of 1 degree Celsius. Her heart was beating probably 2 to 3 times a minute, and she's breathing about twice a minute as well. And this amazing physiological phenomenon has not surprisingly attracted the interests of scientists for decades now. But typically the approach has been to study these animals in a highly controlled situation and go in and take sort of a point estimate of a cold squirrel or a squirrel that's warming up. And these sorts of studies have undoubtedly provided really important information on the underlying physiological and molecular mechanisms of hibernation. But what they don't really allow for is an understanding of the role of hibernation in the natural ecology of the species. So as I mentioned, this animal was hibernating for about 8 to 9 months, so 2/3 to 3/4 of the lives of these animals are spent in this state. So almost certainly it has ecological and evolutionary relevance. And it's this seeing the forest for the tree sort of approach that I'm going to take today in my talk. And so I've switched the axis here to metabolic rate to basically conceptualize the role of hibernation in the natural energetic ecology of the animals. When they're hibernating, their metabolic rate will pretty much mirror their body temperature. Then they're going to wake up, go through various stages of reproduction, and eventually start hibernating again the next fall. So this gives you a bit of an appreciation of traits that are happening during hibernation likely are going to influence how much energy they have to do these more sort of ecologically relevant behaviors. But really what I'm going to focus on today is one specific juncture in a hibernating animal's life, and that is when they emerge from hibernation. So I'm going to address a number of questions in today's talk. The first one is going to be, the first set of objectives, I should say, are going to focus on what are the causes of variation in individual emergence in hibernation. I'll start with the genetic influences on it. Specifically, is emergent state from hibernation a heritable trait? Does it have some evolutionary potential? And secondly, I'm going to ask, what is the association, both at a phenotypic and at a genetic level, between emergent state and reproductive chronology? I'm going to work under a hypothesis that animals are emerging so as to subsequently synchronize their reproduction. And an underlying assumption of that hypothesis is that there's a genetic linkage between the two traits. For evolution on one trait, or pardon me, selection acting on one trait, reproductive phenology, to induce an evolutionary response in another trait, the two need to be linked genetically. And then I'm going to move and start to talk about the environmental influences on hibernation phenology. Specifically, how is emergent state influenced by the weather variables that the animals are exposed to? And also, have any relevant weather variables shown any temporal trend over the course of our study? Once I've established the causes of variation in phenology, I'm going to move into the consequences. Specifically, how does when an individual animal emerges from hibernation, how does when they emerge influence their individual fitness? And across the population, how does it influence the dynamics within the population? A main contention of my talk, or sort of running theme, throughout it is that going to be to truly integrate animal physiology and evolutionary ecology, which is one of the main goals of my research program. We need to start studying animals in more diverse environments than this one. This is not to take anything away at all from physiological or lab-based physiological studies, which have undoubtedly provided important information on the underlying molecular mechanisms and physiological mechanisms of hibernation. What they don't allow for, though, however, is a true understanding of the integration of physiological traits within the ecology of the natural ecology of the animals. As well, I mean, until relatively recently, technological limitations limited the study of many physiological traits to these sorts of environments. But technological advances have allowed us now to take that sort of measurement into more field-based conditions. So we can measure body temperatures of a hibernating ground squirrel using the data logging collars that I showed on the first slide. We can use portable respirometry to measure the metabolic rates of animals exposed to natural conditions. And if we do that in studies in which we can link it to detailed information on the life history, ecology, and population ecology of the animals, we should be able to reach a level of integration between these two fields that you simply can't reach in a purely lab-based study. To begin to understand what has traditionally been considered a physiological trait, hibernation, within the natural ecology of the animals, sort of allows or essentially requires us to imagine it as a phenological trait. Most environments, north-temperate environments, or most environments throughout the globe, exhibit a seasonality. These are the average temperatures for Lubbock. They exhibit the same sort of seasonality that we see throughout much of North America. Where I study the ground squirrels would show a very similar curve, albeit with a slightly more reduced intercept. But basically what it's showing is typically an energetically favorable period is bounded by energetically unfavorable periods. The summer bounded by the winter. And what the challenge to wild populations then is to try and synchronize their reproduction to maximize the benefits, the energetic benefits from these energetic peaks. We can also take a more physiological approach to the study of phenology or hibernation if we think about the energetic costs of staying warm to these animals. So what I've shown here on the graph is a typical metabolic response to temperature curve for an endotherm, that is a bird or a mammal. And I show the typical thing where within a range of temperatures we call the thermal neutral zone, which is right around the range of the temperatures of the room that we're sitting in right now. Metabolic rate is relatively low and stable, and if some other assumptions are met, such as the animal's at rest and it's not digesting, we would call this the basal metabolic rate, or BMR, of the animal. But what starts to happen is as ambient temperature decreases, metabolic rate needs to start increasing. So whereas on the one hand, the ability to maintain a warm, constant body temperature has allowed these animals to inhabit some of the coldest areas of the globe, the ability to do that, to keep that constant warm body temperature, comes at tremendous energetic costs, both in terms of staying warm when it's cold, but also in terms of maintaining that metabolic machinery. The basal metabolic rate of a mammal, for example, is 12 to 20 times higher than a similarly sized reptile. And at the same time, as it's costing more to stay warm, the energy that you can accrue from the environment is going down. As temperatures start to decline, the energy resources that can be obtained from the environment as well start to decline. Plants start to senesce, insects become inactive, etc. Once we put these two things together, essentially the animals are working within a red or energy deficit budget. And maintenance within this period just can't be sustained for any length of time. So an animal basically needs to write the equation. There are two ways to do that, essentially. An animal can move geographically to a more energetically suitable climate. And of course, migration is observed in lots of things like migratory birds, monarch butterflies, bats, for example. But for more sedentary animals, however, such large-scale movements just simply aren't feasible. And really the only way to write the equation is to do so in situ. And that is by entering a state of dormancy, these animals can actually reverse the response between temperature and metabolic rate. Whereas to stay warm, it costs a lot of energy to stay warm and increase your metabolic rate. If you allow your body temperature to drop, the metabolic rate actually goes down with temperature because enzymatic activity slows down. And hibernation, mammalian hibernation, is a commonly observed form of winter dormancy in many species. It's relatively widespread. It's been observed in 7 orders of mammals, everyone with a little H beside it here, including some primates. Hibernation itself, the term comes from the Latin, which means to pass the winter, and is typically used for periods of weeks to months in winter times to accrue energetic savings. Among a suite of phenotypic traits, hibernation is characterized probably predominantly by pronounced behavioral and physiological depression. The metabolic rate of a hibernating ground squirrel is about 5% of the metabolic rate of an animal at BMR, and less than 1% of the animal should it be trying to stay warm at that same ambient temperature. These metabolic savings are obtained by allowing body temperatures to drop to incredibly low levels. Body temperatures at 0 degrees have been recorded for many species, and body temperatures below 0 degrees have been recorded from some mammals, the Arctic ground squirrels up in Alaska. So I've been studying hibernation phenology in a natural population of Columbian ground squirrels up in Alberta in the Rocky Mountains. Some of the natural history of this species is they range throughout the Northwest Rockies, sort of throughout Idaho, Washington, BC, Alberta, etc. They're a herbivorous animal, and they tend to live in alpine meadows, floodplains. More recently, agriculture is providing habitat for them. Relatively long-lived for a small mammal. Some animals can live up to 12 or 14 years. On average, it's about 4. This allows us to get hibernation emergent states multiple times for the same animal. They have a single day of estrus, so the females will wake up and like clockwork, they'll enter estrus 3 or 4 days later, after which they'll have 1 litter per year, approximately 3 offspring, anywhere between 1 and 6. It's hard to give them a mass. Basically, they're that big. And the problem is that they have huge mass cycles throughout the active season. They'll emerge at their smallest mass, and they'll essentially double that by the time they go into hibernation the next a couple months later. Direct relevance, or most direct relevance for our talk, is they're obligate hibernators. Every individual in every population hibernates. The animals will start to emerge about a month, almost exactly a month from now. As I mentioned, the females will enter estrus 3 or 4 days later. They're then pregnant for 3.5 weeks. They're then nursing their babies for about 4 weeks, which essentially leaves them about 6 weeks to pack on all that energy, half again of their body weight, to survive the following 8 to 9 months of hibernation. Suggesting that such a tight cycle, any sort of monkeying around in this area could have some ramifications for the ecology of the animals. The study itself was initiated by Jan Muri, who's now emeritus at the University of Alberta. As I mentioned, it's in an area of Alberta we call Kananaskis. It's a fully census population. The population itself, I showed a picture of on the previous slide, so 3 football fields or so in size. On one side we have a river gorge, and on 3 others we have forest, which the animals don't like to go to. So essentially we have a closed population, which allows us to get phenology, reproductive success, and survival for every animal in the population every year. Basically what we end up doing is showing up to the field site. We'll head out in about 3 or 4 weeks, start sitting up on these towers that are about 10 feet in height, and that allows us to survey the entire population. Once every animal emerges, they all have these little ear tags. You can't see too well, they're little earrings that have an alphanumeric code on them. It allows us to link the individual to the data set and to the pedigree. They'll get a little dye mark on their back so we can tell who everybody is without having to trap them every time. And they're peanut butter addicts, so it makes it pretty easy to trap them. And record their behavior just through behavioral observation. Overall, the data set after those 20 years is about 1,200 individuals deep, recorded for a period of about 20 years. So the most fundamental ingredient for all of my work, and indeed one of the most fundamental ingredients for evolutionary change, is individual variation in the phenotypic trait of interest, which surprisingly, shockingly, is only known for a handful of hibernators. So the first thing that I had to do is say, how much variation is there in when the animals are emerging from hibernation? And this shows that data for the female segment of the population. First thing that showed up is there is annual variation. So the earliest year on record, the average female showed up about 3 weeks earlier than the latest year on record. So all of this data is standardized by annual averages and shows the way